These are the lecture notes for Chapter 13 on meiosis. We just covered Chapter 12 on mitosis, and now it's Chapter 13 on meiosis. I didn't mean to act like this beautiful family wasn't important. They are showing this picture just because the meiosis is related to sexual reproduction. These two children, I'm assuming they appear to be the two biological children of the couple here. So both of these children were created by his sperm and her egg uniting. That's a process called fertilization. They both started out as a zygote, which is a single cell, fertilized cell. And then that zygote divided many, many, many times until, they became born, and then they became this age. But anyway, that's the only way. Meiosis, the purpose of meiosis is for sexual reproduction. It is to create sperm and egg so that you can create offspring. And this illustration on the left, where the chromosomes are shown in blue, this is representing what happens in the father's testes to the parent cell when it is dividing to become sperm. So the parent cell it's a sex cell, a stem cell that only produces sperm, okay? But the parent cell has 46 chromosomes, and the sperm are only going to have 23. So basically that's what happens is when the gametes form, whether it's sperm or egg, when the gametes form, they're going to only have half of the DNA. If the parent cell has 2 chromosomes, each of the sperm and each egg will have only 1 chromosome. If the parent cell has 46, then the sperm and the eggs will have only 23. So the sperm and the eggs, or the gametes, are only going to have half of the DNA, or half the number of chromosomes of the parent cell. And another thing about meiosis you may have noticed is there are 2 divisions. The parent cell divides, well, first of all, here's your parent cell. The chromosomes duplicate during interphase. And then you have the division of that parent cell one time. So you get 2 daughter cells, and then those 2 daughter cells divide and produce 4 daughter cells. So meiosis produces 4 daughter cells. Either they're going to be called sperm or eggs, depending on if they're formed in a female or a male. But meiosis produces 4 daughter cells that only have half of the number of chromosomes of the parent cell. And I will tell you this too, the first division is called meiosis 1. And during that division, the overall purpose is, it says homologs are separated. So I'm going to say homologous pairs are separated. So these 2 chromosomes here are called homologous pairs of chromosomes. And what that means is these homologous pairs of chromosomes, one came from his father and the other came from his mother. So in other words, one came from the grandfather, one came from the grandmother. So this homologous pair of chromosomes has genes for the same traits. Now they might not be the same versions. Maybe there's a gene for green eyes and then a gene for blue eyes on the other one. But they're going to be genes for the same traits. And during meiosis 1, only one of those sets of homologous chromosomes will end up in the 2 daughter cells, that's produced. And then when they divide, the sister chromatids separate. So in meiosis 2, the sister chromatids separate. And that may sound familiar, and it should, because meiosis 2 really is just meiosis. Meiosis 2 is just mitosis. So one of these sperm is going to fertilize one of these eggs, and that will form a zygote, which will end up having the same number of chromosomes as the parent cell up here and the parent cell here. The same number of chromosomes that you would find in every human body cell. So now the fertilization restores the chromosome number. It was halved in meiosis. Now it's going to be restored. All right. A few terms that you need to understand, in this chapter. Heredity just means the transmission of traits from one generation to the next. In sexual reproduction, sons and daughters are not identical copies of either parent or of their siblings. So along with similarity that they inherit because they have, the same parents, they're also going to have variation. You've probably seen siblings that look alike and you can tell that they're siblings. And then they also favor their parents. But you can probably also think of a situation where a biological child doesn't look like any of its brothers or sisters, nor either parent. And that's because of genetic variation. That's because sometimes it just works out that way, that their traits, the combination of traits they inherited, they just don't end up looking, similar to the rest of their family. That happens sometimes. Now, the word heredity is the passing of traits from one generation to the next, but the word genetics is the study of heredity. So heredity is the passage of traits from one generation to the next. Genetics is study of heredity. And then a gene is defined as a unit of heredity. In other words, it is a piece of DNA or a segment of DNA located on a chromosome that codes for a trait. So we say a gene is a unit of heredity because one gene will code for one trait. And genes are passed to the next generation through the gametes, through the sperm and the eggs. DNA is packaged into chromosomes, so chromosomes are going to be DNA plus protein. Humans have 46 chromosomes in their body cells or their somatic cells, and then they have 23 in their gametes. The place, by the way, this is another term that you need to know in bold. The place or the position along a chromosome where a gene is located is called its locus. And if we say the location of more than one gene, we say loci. Now, in asexual reproduction, this means that you don't have sperm and egg, you don't have variation. All of the offspring are literally clones of the parent. But in sexual reproduction, you have two parents, you have two different gametes, a sperm and an egg, or like in the case of fungi have a positive and a negative version. You don't really call them sperm and egg, but it works the same way. So in sexual reproduction, you have a mixture of traits inherited from each parent. So each parent gives half of the DNA, of an offspring's DNA, to the offspring. And you can see this is an example of cloning in Hydra or asexual reproduction. Redwoods are actually all clones of one another, and they're pretty much joined underground. It's almost like they're one organism. This is a video of the Hydra budding, which is basically you have the Hydra is like a jellyfish, but it's turned upside down compared to a jellyfish. It has tentacles at the top instead of tentacles, pointing downward. So here's the parent with its tentacles, the parent Hydra, and then a little baby bud forms off of the side of the parent's body, and then that bud will break off and it will attach somewhere. The Hydra live in fresh water. So it'll attach somewhere and it'll get bigger and it will become an adult Hydra. A life cycle is the generation to generation sequence of stages in the reproductive history of an organism. So we talk about asexual life cycles and sexual life cycles. I'm going to stick with, there's a lot of information and comparisons among like the life cycles of a plant versus a fungus versus a human. And I'm going to stick to the human life cycle in chapter 13 because I think that's enough, for you to have to learn. When you get to Bio 112, you can go into more detail about other organisms, but we're going to stick with humans when we learn meiosis. So, human somatic cells. Remember, somatic cells are body cells. They're going to be all the cells that are not sperm and egg. All of our human somatic cells have 23 pairs of chromosomes, and 23 pairs, of course, is 46. All right, we can see all 46 chromosomes in a photograph called a karyotype because you can see them under the light microscope and you can take a picture of them. During cell division, the chromatin condenses and forms chromosomes. So what you do, you take a picture of the chromosomes and then you use a computer program to line them up, match them up with their homologous pair, and then, line them up, and you can use a karyotype to determine if, like, the baby is going to have a chromosomal abnormality, such as Down syndrome. You can also use it to tell the biological gender or sex of the baby. All right, but they don't usually do that. I mean, they do that with an ultrasound. But anyway, I told you about homologous chromosomes. You're going to see the word homologs, but I usually just say homologous chromosomes. So since there's 23 pairs of chromosomes in humans, that means there's 23 pairs of homologous chromosomes. 23 homologous pairs is what we say. And the way that you know which two chromosomes are in a pair, in a homologous pair, they're going to have the same length. They're going to be the same height. They're going to have the centromere, or the waist, will be in the same position. And their staining pattern, which kind of represents their staining or banding pattern, shows you their genes. So the staining pattern is the same, which means they have genes for the same traits. So here's a karyotype, and you can see that all the homologous pairs are lined up all the way to pair 22. This last pair is pair 23. And you can see one of the chromosomes is an X, the other is a Y. So that tells us that this is the karyotype for a male. And you can also tell that this male does not have a chromosomal abnormality because it's got 2 chromosomes for each pair. It's not missing any chromosomes, and it doesn't have any extra chromosomes. And like I just said, so there are 22 pairs of chromosomes called autosomes. And they don't have anything to do with determining the biological sex of the baby. But then there are the 23rd pair are the sex chromosomes. So the sex chromosomes in a human female are going to be 2 X's. And then in a human male, there's going to be 1 X and 1 Y. And it's actually the presence of the Y that makes the human male a male, the presence of that Y chromosome. We use the term diploid, and we represent it with 2N, the symbol 2N. That represents cells that have 46 chromosomes, or 23 pairs. And then we use the term haploid because the diploid number in humans is 46. And then in the gametes, we use the term haploid, which is just an N, and it's 23 in humans. that will represent the chromosomes in the gametes, or the sperm and the egg. They're going to have the haploid number, and all the other cells, all the other somatic cells, are going to have the diploid number of chromosomes. And then fertilization, of course, is the union of the sperm and the egg. And then the fertilized egg, we call that the zygote. And then the zygote, which is the sperm and the egg, are produced by meiosis. And then fertilization gives you a zygote. But when the zygote begins to divide and form, trillions of cells, that's going to be mitosis. And this is an illustration of the life cycle of a human. So we'll just start with fertilization. So you have an egg that has 23 chromosomes, the haploid number, a sperm that has 23 chromosomes, the haploid number. The sperm unites with the egg, that's fertilization, and now the zygote is diploid. And that zygote divides by mitosis and produces the adults. In the testes of the adult male, meiosis occurs and produces a sperm. In the ovaries of an adult female, meiosis occurs and produces egg. And then the process just continues. The sperm and the egg are haploid. All the other body cells are diploid. So gametes are only produced by meiosis. And it occurs in a female, a human female. Meiosis occurs in the ovaries. And in a male, meiosis occurs in the testes. Now, here is where they compare plants and fungi with animals. And you can read over that if you want to. I'm not going to ask any questions about it. All right, now we're going to talk about meiosis 1 and meiosis 2. But I do want you to know meiosis, you do have the replication of or duplication of chromosomes before meiosis begins. But then meiosis has two cell divisions called meiosis 1 and meiosis 2. And at the end of meiosis, you get 4 daughter cells instead of 2, and they're haploid. They only have half the number of chromosomes as the parent. So this is what it looks like. You've got a parent cell. Before meiosis begins, the chromosomes in the parent cell are duplicated. In meiosis 1, so what we're looking at here is one pair of homologous chromosomes. So in meiosis 1, the homologous chromosome pairs separate, and each daughter cell that's produced gets one copy. Then in meiosis 2, the sister chromatids separate. So you end up with 4 cells that have only half the number of chromosomes of the parent. All right, there are 4 phases of meiosis, and depending on if it's meiosis 1 or meiosis 2, we use prophase 1, metaphase 1, anaphase 1, and telophase 1 for meiosis 1, and then we use Roman numeral 2 if it's meiosis 2. Now, I'm going to stop here. This is slide number 33. So I will start with slide 34 in the next recording.